Welcome. Today, we're diving into the fascinating world of fish biology, focusing on how age impacts a fish's ability to survive in changing salinity environments. Osmoregulation is the process by which organisms maintain the balance of water and salts in their bodies, a crucial function for all living things, especially for fish that move between fresh and salt water. As fish age, their ability to regulate these internal balances can decline, affecting their survival. In this video, we'll explore recent research on Mozambique tilapia, a Euryhaline fish known for its remarkable salinity tolerance, and see how age changes their osmoregulatory abilities. Uraheline fish, like Mozambique tilapia, are unique because they can tolerate a wide range of salinities. This means they can move from freshwater to seawater and vice versa, adjusting their internal processes to survive. This adaptability is essential for species living in environments where salinity changes frequently, such as estuaries or tidal zones. However, not all fish can do this, and even among Euryhaline species, the ability to adapt can change with age. Understanding Euryhalinity helps us appreciate the challenges these fish face and sets the stage for our discussion on aging and osmoregulation. Osmoregulation involves a complex interplay between cellular sensors, hormones, and specialized tissues. In fish, the gills play a central role, acting as the main site for ion exchange with the environment. Hormones like prolactin and various transport proteins help regulate the movement of ions such as sodium and chloride, ensuring the fish maintains proper internal conditions. As fish age, these processes can become less efficient, leading to challenges in maintaining homeostasis, especially during rapid changes in environmental salinity. Studying aging in fish provides valuable insights not only for aquatic biology, but also for understanding similar processes in humans. As vertebrates age, their ability to maintain water and salt balance declines, leading to health issues. Fish offer a unique model because their gills function similarly to human kidneys in regulating ions. By examining how aging affects osmoregulation in fish, scientists can uncover mechanisms that may be relevant across species, including humans. Mozambique tilapia, or Euryachromis mozambicus, are ideal for studying osmoregulation due to their natural exposure to fluctuating salinities. These fish have evolved mechanisms to survive both in freshwater and seawater, making them perfect subjects for experiments on salinity tolerance and aging. Researchers can observe how their physiological responses change over time, providing a window into the aging process in aquatic environments. Researchers designed experiments to test how age affects tilapia's ability to survive sudden changes from freshwater to seawater. Fish of different ages, 1, 4, 6, and over 24 months, were transferred from freshwater to seawater, and their survival was monitored. Some groups experienced rapid transfer, while others had a gradual transition. This setup allowed scientists to compare how quickly and effectively young versus old fish could acclimate to new salinity conditions. The results were striking. Younger tilapia had much higher survival rates after transfer to seawater compared to older fish. All 1-month-old fish survived, while only 55% of 4-month-olds and 15% of 6-month-olds made it through. Notably, none of the fish older than 24 months survived the rapid transfer. These findings highlight a clear age-dependent decline in salinity tolerance, suggesting that older fish are less capable of handling sudden environmental changes. When the transfer from freshwater to seawater was done gradually, survival rates improved for all age groups. For example, 4-month-old tilapia had 0% survival with a 1-hour transfer, but 70% survived with a 3-hour transfer, and 100% with a 12-hour transfer. Even among the oldest fish, a slow transition significantly increased survival, though not to the levels seen in younger fish. This demonstrates that giving fish more time to acclimate can partially offset the effects of aging. To understand why older fish struggle, researchers examined the expression of key ion transporter genes in the gills, NCC2, NKCC1A, and NK-alpha-1A. These genes help move ions in and out of the fish's body, crucial for adapting to new salinity levels. In younger fish, these genes responded strongly to changes in salinity, especially NKCC1A, which was significantly upregulated after seawater transfer. In older fish, this response was much weaker, indicating reduced sensitivity and adaptability. Prolactin is a hormone well known for its role in osmoregulation in fish. It helps fish absorb ions from their environment, especially in freshwater. In this study, researchers looked at how prolactin-producing cells in the pituitary gland responded to changes in osmolality. They found that cells from young fish released more prolactin when exposed to low osmolality, while cells from older fish showed little to no response. This suggests that aging reduces the sensitivity of prolactin cells, impacting the fish's ability to adapt. To further test prolactin's effects, scientists incubated gill filaments from both young and old tilapia with different concentrations of prolactin. In young fish, the expression of the NCC2 gene increased significantly with higher prolactin doses, showing a robust response. In contrast, older fish had a much weaker response, even at the highest prolactin concentration. This experiment confirms that aging diminishes the gill's sensitivity to hormonal signals, further compromising osmoregulation. Osmorception is the process by which cells detect changes in the surrounding water's salt concentration. This triggers hormonal and cellular responses to maintain balance. The study found that osmorception itself becomes less effective with age, meaning older fish are slower or less able to sense and respond to salinity changes. This decline in sensory function is a key reason why older tilapia struggle with rapid environmental shifts. Interestingly, the decline in osmoregulatory capacity with age seen in fish mirrors similar processes in humans. As people age, their ability to maintain water and salt balance also decreases, leading to issues like dehydration. By studying fish, researchers can gain insights into the molecular and hormonal changes that occur with aging, potentially informing medical research on age-related homeostatic decline in humans. Let's briefly recap the experimental methods. Researchers used controlled tanks to expose tilapia of different ages to rapid and gradual salinity changes. They monitored survival, sampled gill tissues, and measured gene expression using quantitative PCR. They also isolated prolactin cells and tested their response to osmotic changes in vitro. This comprehensive approach allowed for a detailed understanding of how aging affects each step of osmoregulation. To ensure the results were robust, scientists used statistical tests like two-way ANOVA and t-tests to analyze gene expression and survival data. They compared groups by age and treatment, looking for significant differences. This rigorous analysis confirmed that age and salinity both had strong effects on survival and gene expression, with clear interactions between the two factors. The key takeaway from this research is that age significantly reduces a fish's ability to survive sudden increases in environmental salinity. This is due to a combination of reduced gene expression in osmoregulatory tissues, decreased hormonal sensitivity, and impaired osmoreception. These changes collectively undermine the fish's ability to maintain internal balance, making older individuals more vulnerable. These findings have practical implications for aquaculture and fisheries management. Understanding that older fish are less tolerant of rapid salinity changes can inform best practices for moving or acclimating fish in commercial settings. Gradual acclimation protocols can help improve survival rates, especially for older stock, ensuring healthier populations and more sustainable operations. The study opens up new questions for future research. For example, could differences in body size within the same age group also affect osmoregulatory capacity? Are there ways to enhance the resilience of older fish through diet, genetics, or environmental conditioning? Exploring these questions could lead to new strategies for improving fish health and understanding aging in other animals. It's important to note that all animal experiments in this study were conducted in accordance with ethical guidelines and approved by the relevant institutional committee. The data from this research are available in public repositories, ensuring transparency and enabling further analysis by the scientific community. To sum up, aging in Mozambique tilapia leads to a decline in their ability to survive rapid salinity changes. This is due to reduced gene expression in gill ion transporters, diminished hormonal responses, and weakened osmorception. Gradual acclimation can help, but older fish remain at a disadvantage. These findings not only inform fish biology and aquaculture, but also provide insights into aging and homeostasis in other vertebrates. Thank you for joining us on this exploration of aging and osmoregulation in fish. We've seen how age impacts survival, gene expression, and hormonal sensitivity in Mozambique tilapia. These findings highlight the importance of considering age in both research and practical applications, from aquaculture to broader studies of aging. Stay curious and keep exploring the wonders of biology.